Thanks for the invitation for us to speak here today. Today, Christina and I will be presenting our pooled prime editing platform for high throughput interrogation of variant effects. And so to start by saying that as genomic sequencing is becoming more and more commonplace in clinical practice, our databases of human genomic variation is growing rapidly. To take full advantage of the wealth of available genomic data, it is critical for us to understand how these variants can affect human health. And so currently, the effects of most observed human variants are not fully understood, which is illustrated here using recent data from ClinVar. These variants of unknown clinical significance have also seen a stark increase over the last few years. And so for optimal patient care, as well as to illuminate disease mechanisms and develop better therapeutics, it is essential for us to bridge this gap between genomic and functional data. And so there are many different methods for us available to efficiently link genotype to phenotype at scale. Some of these include human genetics, as well as computational predictors and functional assays. In our lab, we're predominantly focused on functional assays, in particular on multiplexed assays of variant effects, or MAFs. So MAFs are high-throughput methods to interrogate the functional effects of thousands of variants in a single experiment. Phenotyping then usually is based on the measuring out variant abundance changes in the assay using next-generation sequencing. Even so, there's a wide array of different MAF formats, but we believe that the ideal platform should satisfy the following three criteria. First, it should enable highly scalable experimental throughput, should also enable the testing of diverse variants. And finally, it should also enable the testing of these variants within the native genomic context. And so current technologies satisfy some of these criteria, but not all of them. And so, for example, some of the more traditional MAF formats, such as deep mutational scanning and massively parallel reporter assays, are highly scalable methods that can test diverse variants. But because variant libraries are usually delivered exogenously, so often on plasmids, the genomic context is completely missing. On the other hand, with methods such as saturation genome editing or SGE, variants are encoded on donor plasmids and delivered to cells, where they then serve as donors for HDR-mediated integration into a desired genomic locus using Cas9. And so this enables the installation and testing of diverse variants directly in their genomic context. However, unlike DMS and MPRAs, scalability is much reduced, and that comes from the fact that variant effects are determined from sequencing out variants at their endogenous locus. So in practice, SG experiments are confined to a small genomic region of around 200 base pairs. For cooled CRISPR screens, finally, which there are formats for nuclease-based knockout screening as well as base editing screening currently. For these assays, guide RNA libraries are delivered to cells in lentiviral libraries, which then enables the installation of variants genome-wide. Scalability is much improved compared to SGE, for example, because variant effects are determined directly from guide RNA abundance changes and not the changes on endogenous level. However, tested variants are currently limited to either indels or very specific point mutations on the basis of currently available CRISPR tools. And so we reasoned that employing prime editing within a pooled screening platform would enable a method that satisfies all three of these criteria. So prime editing is one of the more recent genome editing tools developed in 2019 by the David Liu lab. It enables direct DNA writing into the genome. And it achieves this using a two-component system consisting of a prime editor as well as a prime editing guide RNA or pegRNA. The prime editor itself consists of a Cas9 nickase fused to a reverse transcriptase. And the pegRNA is a traditional guide RNA with a 3' extension which encodes the desired edit, which then serves as a template in the reverse transcriptase activity of the prime editor. Prime editing is a very powerful genome editing tool, in part because it enables the installation of very diverse edits, such as all possible single nucleotide variants, as well as smaller insertions and deletions. And it also achieves this at very high precisions, meaning usually minimal byproducts are observed with the desired edit. And so in part because of this, as well as the fact that pegRNAs specify genomic target site and desired edit, it's also a highly scalable genome editing tool. So as I said, we set out to establish a prime editing platform for high throughput variant screening, similar to currently available nuclease-based knockout screens and base editing screens. And so I just want to mention here that various other groups have also established similar platforms around the same time that we did. And the known comprehensive list of these are listed on this slide. However, the fundamental steps of the screening procedures generally shared across all of these. And they include first the computational prioritization of a set of variants, and then the design of a corresponding pegRNA library. This pegRNA library is then synthesized and cloned into a vector for lentiviral packaging. The resulting lentiviral pool is then used for the transduction of cells already expressing a prime editor. And as the prime editor and pegRNA are complemented within each cell, prime editing activity can begin and variants are installed across the genome. This then follows a cell selection step where these variants are challenged in the cell pool. And finally, we conclude by sequencing the lentivirally delivered pegRNA cassettes to then determine variant effects as a measure of pegRNA abundance changes across the cell selection step. And so going into this, we anticipated that one of the biggest challenges in setting up a successful pooled prime editing platform would be achieving sufficient prime editing efficiency. And so we ensured to implement all possible prime editing optimizations to achieve sufficient prime editing efficiency for screening. And so the first step for this was using engineered pegRNAs or ePegRNAs. These are pegRNAs with an additional 3' cap. We used TiVo-preQ1. This both stabilizes the pegRNA as well as protects the pegRNA against any nucleases. For the host of our platform, we have chosen near haploid human cells called hap1. These are widely used cell lines in the functional genomics in general, but in high throughput screening especially. We reasoned that their haploid status would best facilitate both the editing as well as variant interpretation. Within these hap1 cells, we installed the most efficient prime editor at the time called PMax. And from that same cassette, we're also co-expressing dominant negative MLH1 protein or MLH1DN. This serves to inhibit the cell's DNA mismatch repair pathway, which, if left intact, would revert some of the prime editing outcomes, thereby overall reducing overall editing efficiencies. We also implemented a co-selection step or edit enrichment step in the particular system that we implemented. Together with every variant of interest, we're also installing a variant within the ATP1A1 gene. This specific ATP1A1 mutant then confers resistance to a drug called uabine. And so using this strategy, we can enrich cells within the cell pool that have both edits present in the genome using a simple drug treatment. Finally, despite all of these optimizations, we reasoned that primer editing efficiency will depend heavily on each peg-RNA, and there will be high variability there. So we also included a surrogate target together with each peg-RNA. These surrogate targets or sensors are small copies of the endogenous target of each peg-RNA and are read out together with the peg-RNA in the final sequencing at the end of the workflow. So they enable the monitoring of each peg-RNA's activity throughout the screen. This strategy has been used before to track CRISPR editing outcomes, and more recently also within pooled base editing and prime editing screens. Okay, so at this point, I want to give over to Christina, who will be diving into the actual screens we performed as part of this work.
Okay, so to establish general feasibility of our method, we have performed a series of proof of principle experiments. And today we want to share the results of 2 of them, which both explore distinct potential application for our platform. In the first experiment, our goal was to target variants in a gene that is essential in HAP1 using a negative selection framework. This means that loss of function variants lead to cell death and will be depleted throughout the experiment. We chose to explore this because one unique feature of HAP1 cells is that there are more than 2,000 such essential genes, which could all become accessible in future screens. The gene we chose was SMARCB1. This is a chromatin remodeler implicated in the development of different diseases, including tumor predisposition syndrome as well as developmental syndromes. It is commonly sequenced in the clinic, and currently about 45% of the known variants are of uncertain significance, such that functional data could be immediately valuable. And the variants we selected for this experiment were all of the possible point mutations across 2 exons, 8 and 9, which were located at the C-terminal domain. And in total, these were more than 1,300 variants. As Michael pointed out, we anticipated that there may be challenges with editing efficiencies, which is why we designed several guides for each of the variants we tested. We then introduced our library containing more than 10,000 guides into a pool of HAP1 cells and selected samples at several time points before sequencing the stably integrated constructs. And one of the first questions we addressed was what are the editing efficiencies we actually achieved? And as you can see here in the plot, edits continue to accumulate throughout the experiment. And this can be explained because both the prime editor as well as the guides are constitutively expressed. Importantly, our Urban enrichment strategy turned out to be successful, and we benefited from an additional small boost here. So on our final time point, day 34 in this experiment, we reached a median of about 40% editing at the surrogate target. It becomes quite apparent already here from these plots that our concern of widespread ranges in terms of editing were warranted, because we can see that activity differed quite a lot throughout the pool. We then went on to address the really key question of this experiment, which was, can we infer variant effects by only looking at the changes in peg-RNA abundance without having to sequence the endogenous sites where the variants are installed? And to this end, we calculated function scores by comparing the peg-RNA frequencies at the experimental endpoint to an earlier time point, which you can see here plotted by grouped by the molecular consequences of the corresponding variants. And because in this experiment, loss of function variants will be depleted throughout the experiment, they are expected to receive a high score. And in this plot, I want you to focus your attention on the first 3 variant groups, which served as the controls in this experiment. Synonymous variants are expected to be neutral, while nonsense and canonical splice variants are expected to be depleted. As you can see, if we include all of the guides we have designed into this analysis, we are not able to resolve these expected differences. And this might not be surprising, considering that we see this widespread in editing efficiencies. However, this is really where the surrogate target comes into play, because monitoring the activity of all the guides allows us to repeat this analysis, retaining only the most active guides. And then the picture already becomes quite different. So now we are able to resolve the differences between the control groups. And on top of that, we can also identify loss of function variants across the additional variant groups. So overall, this experiment taught us that using our platform in the context of negative selection is possible. However, it is really dependent on achieving high editing efficiencies. And in this particular experiment, accurately resolving variant effects came at the cost of reduced coverage. We then moved on to our second experiment, switching gears a bit. Here we used a positive selection framework because we reasoned that we could maybe overcome some of the editing-related challenges by choosing an assay that has a stronger phenotypic effect size. The gene we targeted here was MLH1, which is one of the core mismatch repair genes. And Michael has already briefly touched on this gene in the context of optimizing our platform. However, it's in its own right also a quite interesting gene because pathogenic variants in MLH1 cause one of the most common tumor predisposition syndromes called Lynch syndrome. Again, as for SMARCB1, there's a large proportion of variants of uncertain significance currently reported for MLH1. Conveniently to us, if HAP1 cells lose MLH1 function, they become resistant to a drug called 6-duraguanine, which we could use as the functional readout in our experiment. In this experiment, we now also wanted to expand the scope of the types of variants we tested from just coding variants to non-coding variants, which are a class of variants that have historically been quite challenging to test at scale. We selected all of the variants that at the time were present in the ClinVar database, which you can see here spread across the full gene body, marked as blue ticks. Many of these variants are located close to exon-intron boundaries, where clinical sequencing usually takes place, but some variants were also found deeper within introns. And we're really excited about this experiment because targeting so many diverse non-coding variants spread across a large genomic region was just not possible with any other screening method. If we now look at the data we generated, this is a gene function map. So on the x-axis, we have the genomic position of all of the variants we have tested. And on the y-axis, we again have a function score, which we derived by comparing the frequencies of peg after selection to the frequencies before selection. And because in this experiment, loss of function variants will be enriched through our drug treatment, they will receive a high score. And again, I want you to focus on the control groups, which here are the variants colored in yellow. On the one hand, these are canonical splice variants that are expected to be deleterious. And on the other hand, we have variants in light blue, which are intronic variants outside of splice sites, which predominantly are neutral. And we can see that we achieve quite a nice separation of these two groups, which importantly was achieved by only very moderately filtering our data set based on editing efficiencies. So here we were able to retain about 50% of the variance in analysis compared to just 20% in the negative selection screen. Because most of these non-coding variants that score highly are expected to have an effect on splicing, we then compared our function scores to predictions from Splice AI. And we found that these correlated quite well, which further increased the confidence in our results. Since all of the variants we tested were taken from ClinVar, we also had the opportunity to compare our scores to the pathogenicity annotations in the database. And this analysis also supported our scores. Most pathogenic variants are scoring highly, while benign variants generally score lowly. We can see that if we apply a moderate filter, the score ranges are still overlapping. However, here we can again briefly touch on the ability to tune the accuracy of our results by applying more stringent filters. If we do this, we can completely separate benign and pathogenic variants. We now also want to highlight 3 concrete hits that we found in the screen, which illustrate some of the types of discoveries we can make with these experiments. On the one hand, we want to show this 8 base per deletion upstream of exon 1 that extends into the translation initiation site. This variant shows that not all of our hits act via splicing, but in a single experiment, we were actually able to detect multiple loss of function mechanisms. On top of this, deletions are also among the variant types that we could not have screened with base editing screens. The second hit we want to point out is the single nucleotide variant within intron 16. This variant is more than 200 base pairs away from the nearest exon boundary, and one of the few known deep intronic variants that causes disease in humans. We think it's quite encouraging that our platform was able to pick up on its effect, because it suggests to us that the platform could in the future be used to more systematically interrogate deep intronic spaces and find additional variants like this one. And then finally, we also want to point out one of the variants of uncertain significance that scored highly in our experiment. We believe that pooled prime-editing screens will in the future, like many other MAVE assays, play an important role in tackling the variant interpretation problem and provide functional data for the reclassification of VOS. However, considering the current stage of the technology, it's important to us to point out that these hits should be thoroughly validated before our data is used clinically. And for this particular variant, we have done this by introducing it individually into cells and quantifying splicing outcomes from RNA. And this experiment validated this hit. So to summarize what we've shown you so far, we have built a new variant screening method based on prime editing that is highly flexible and allows us to perform various types of screens. We have shown you that we can perform negative selection as well as positive selection, that we can test the effects of coding and non-coding variants, and that screens can be confined to a focused region in the genome, but can also include variants that are distributed across larger genomic sequences. We think that pooled prime-editing screens fill an important niche within the greater framework of MAVE assays and overcome some of the limitations of other methods. However, we've also repeatedly now touched on one important limitation of our platform, which is that prime-editing efficiencies can be low and they can also vary between different guides, which in our proof of principle experiment had an effect on the scale. However, there's hope, because prime editing is a very fast-moving field, and since we concluded this work, additional optimizations have become available, which we all now integrated into an upgraded version of our platform. And here we just want to highlight the most recent prime editor P7, new computational prediction tools for better pegRNA designs like Predict2, and an experiment where this is feasible, we found that completely inhibiting the mismatch repair pathway by MLH1 knockout has a stronger effect than co-expressing a dominant negative MLH1. Together, this has allowed us to significantly improve the editing efficiencies we achieve in our screens. On the right in this plot, you can see efficiencies from a recent screen 10 days after pegRNAs have been introduced into cells. And just for comparison, on the left side, you can see the same time point from our negative selection screens. And then finally, we just want to give you a glimpse into the future, because these improved editing efficiencies now allow us to massively scale up the experiments we are performing. And in the lab, we use the scale now to perform larger scale negative as well as positive selection screens. So on the left side, you can see a recent experiment in which more than 100,000 missense variants have been tested across 37 HAP1 essential splice factor genes. And for anyone attending ASHG, Michael is going to present this data there for the first time. And on the right side, you can see how we're expanding on the positive selection framework. This is an ongoing experiment where we're testing the effects of more than 50,000 non-coding variants across the whole mismatch repair pathway. And with this, we want to thank everybody who has contributed to this work, especially Greg, for his guidance and mentorship, and also for the opportunity to work with this really exciting technology. We want to thank all of the great members of the Genome Function Lab, and especially point out Megan, who had originally started the prime editing work in our lab. And then finally, we also want to thank the science technology platforms at the Crick, the Advanced Sequencing Facility, and the Vector Core. We're happy to take questions.
Thank you for Christine and Michael for the great talks. So we have one question in the Q&A from Britt Hansen. So for the MLH screen, in addition to Splice AI, did you also consider comparing your functional scores to Pangolin, as this is known to outperform Splice AI?
And if I take this, yeah, I think that's a very good suggestion. We have not done this for the screen we have performed, so we have just used Splice AI to generally benchmark our results. But yeah, I agree that when it comes to predicting effects of splice disruptive variants, it's good to consider more predictors, not just Splice AI.
Great. We have 2 more questions. Okay, 3 more actually. So from Sujata, are the HAP1 cells here LIG4 knockout as well, in addition to having MLH1DN?
Would you mind
I think it's
repeating?
I think it's pretty technical. So are the 8 HAP1 cells here LIG4 knockout as well, in addition to having the MLH1DN?
No, it's exclusively MH1 knockout where possible for the new screens. And then previously for the older screens, we've just used the dominant negative MH1. No other manipulation of DNA repair pathways though.
Okay. One more question from William. Have you tried other cell types?
Yeah, we actually have, because during the validation, or like we've validated some of our hits also in K562 cells, and we generally found that the effects held up. But we've also seen that if we use cell lines that are diploid or polyploid, that there can be more noise, because it is more important that low editing efficiencies can have a larger impact than HAP1 cells. But it's generally possible. Again, like if you can design highly active guides, this works in other cell lines as well.
Okay, last question. So great talk and word thank you from Matthew. How do you think the sensor system compares to endogenous sequencing as a readout? And how problematic, if at all, is recombination with the sensor approach?
Recombination in general, we've seen to be a problem. I think a lot of this is sequencing based, so we just perform pre-filtering on the reads that we get from NGS, and we find usually around 50% of reads are discarded either because we find recombination or there's some synthesis or sequencing error. We have done a comparison for this SMART-P1 data between endogenous target sequencing efficiencies as well as surrogate target, and generally found like a good correlation between the two. But obviously it's much more useful to have an integrated readout of PEG-RNA efficiencies than to go into the endogenous locus to measure out efficiencies, especially if edits become more scattered across the genome.